Okay, so back to topic 7. We had a few things to talk about. I told you I'd do a little flow chart for you. So I'm going to, I got to come here because that's the microphone. I got to keep running back to the computer. So I'm going to draw a little flow chart with you for the blood agar stuff. Okay, so if you have a piece of paper, you can pop that out in a second. So just a reminder about the blood agar. The blood agar literally has blood in it. It's usually 10 or 15% sheep blood. And the whole idea is to enrich the media for certain pathogenic organisms that need that stuff. A lot of them need iron and whatnot to grow properly. And so it is an enriched media. The other thing about blood agar is it is a differential media in that different organisms can grow in different ways. So this is a slide, and you can see that there's alpha, beta, and gamma hemolysis. And that's what I want to draw a little flow chart for you to show what they are. So you can see that the beta hemolysis, for example, hemolysis means breaking red blood cells open. And you can see the beta hemolysis is the most complete. And you can see that there's kind of these little sort of a ghosty halo around the colonies. And that's very distinct of organisms that do beta hemolysis. So I have a little flow chart I want to do. I'm going to try to shout in the direction of the microphone. So it sounds like I'm shouting, I am. And so let's do this and see if I can get this to draw on the screen. Okay, so new share, and where is it? This one? Nope. Why is this not sharing? Okay. Hopefully everyone can see that. You can see I have streptococci and thrown on some blood agar, and we have 3 options. We have alpha hemolytic, beta hemolytic, and gamma hemolytic. Okay? So where's this pen? So I'm going to start off with the beta hemolytic because it is the most complete hemolysis. So here's my note here. See if I can write on the screen. Uh-oh. Try again. Okay, there we go. So beta hemolytic is clear, right? And so this would be what we call complete hemolysis. Okay, so that's kind of it, except for to know, okay, which organisms is this relevant to? So that's what I want to write down. So these are all Streptococcus, so I'm not going to write out Streptococcus, the full word, I'm just going to use the abbreviation. But we have Streptococcus Pyogenes. Okay, so I can't do that in italics with my handwriting, so there we go. And this is the one that we would call, this group A strep. So group A strep. And so the abbreviation is sometimes GAS, and you might see that on medical documents, right? You know, checkbox for base strep, GAS. And so what does this organism cause? This is the one that's famous for strep throat and many, many other infections. It can cause pneumonia, blood infections, and things like that. So maybe I'll write that down. Strep throat. And I'll put plus because it can make all sorts of other types of infections. We also have another organism. So by the way, Streptococcus pyogenes is important for this class. It's going to come up again and again. The second one I'm adding on here, I'm probably not going to talk about it ever again after today, but I want to put it on here just for completion, right? We also have something called Streptococcus, spelling on this, not even sure how to say it. It's agalactinate or something like that. Tia. And this one is called group B strep. So sorry about my handwriting. It's really hard to write on a screen. It's really not ideal. And so this is, of course, GCS. So this is the one, if you've ever been pregnant, or if you are currently pregnant, this is one that they test you for. And they want to make sure, you know, if you have the organism, it's not necessarily bad for the mother, but it can cause very serious infections in the newborn. And so if you have it, well, I think it's a rectal swab, if I remember correctly. And then they test to see if you have it, and if you have it, then they're going to take extra precautions. And so those precautions might be they're going to give you an IV drip to you know, with antibiotics and to kill the bacterium, just to protect the newborn. So it's can be very serious for newborns, like I said, and so it's taken very seriously in prenatal health. Okay, let's look at alpha hemolytic. Alpha hemolytic. How do I do this? I need to. Trying to figure out how I can slide this over. Maybe I can just shrink it a little bit more. There we go. Okay, I'm going to grab a new color. So alpha hemolytic. So these ones, if you looked at the picture on the PowerPoint, they're often a little bit of greenish in color. Okay, so that's worth it to note. So maybe I will grab the green marker for that. So these are greenish, and these are a partial hemolysis. So which organism falls into this category? This is Streptococcus pneumonia. Streptococcus pneumonia. And of course, this causes pneumonia, right? And it can cause other infections. The thing about these Streptococcus organisms is they're very serious in that they can cause quite a variety of infections. You know, they often have their kind of usual areas, like this one here can usually cause, is actually a usual cause of pneumonia. Something like 60 to 75% of all pneumonias are caused by this particular organism. But it can get in the blood. It can cause other infections. And so this is why Streptococcus are taken seriously. Notice there's no group with this guy. I'm going to talk about that in a minute. I think that's on the next slide. But this grouping of these organisms was done by this guy named Lancefield many years ago. And I have no idea how many strains of Streptococcus there are, probably hundreds. And he only classified something like 30 of them. I don't know why he missed an obvious human pathogen. No idea. I don't know the story behind that. So this was never given a group, right? It's just sometimes it's called a non-Lancefield Streptococcus or something like that. Or we just call it Streptococcus pneumonia. Okay, so one more is the gamma hemolytic. So I'm going to use a new color for that. And this one here, there is no lysis. So no lysis. And so what are we talking about here? We're talking about the Enterococcus. So there's a bunch of Enterococcus organisms. For example, there's one Enterococcus faecalis, which of course is associated with feces. These were formerly known as Streptococcus. They were reclassified, I don't know how many years ago. So it's worth it to say that these are formerly Streptococcus, and they were group D strep. So hopefully you can read that. It says, formerly they were known as group D Streptococcus, and they are known for root canal infections and many more infections. Right? Again, you get them in the blood. That's, that can be very serious. You can get them in the lungs. You can get pneumonia from them, and so on. And now they're known as Enterococcus. I won't be talking about Enterococcus really after today as well. Won't be on any of the tests. So the two to know are these two here. I'll put a star beside them, because these are the ones that are going to come up again and again. So those are the two Streptococcus that you do need to know. Just see there's a note in the chat. So what color are gamma hemolytic, if somebody's asking? Usually it's the color of the organism. Most of them are kind of a creamy color, right? So the colony, and I'll show you the picture again that shows the alpha, beta, and gamma all on one slide. Okay, so everyone good? Have that? Okay, I'll switch back to the PowerPoint. All right, there's the PowerPoint. So you can see the gamma. It just looks like any old bacterial colony. It's kind of a brownish color, maybe not as green as the Streptococcus pneumonia. Again, it depends on the exact organism. So thought I had some slides here. Hold on a second. Let me just check something here. Sorry about that. All right, yeah, just figuring out the technology. Okay, so I told you I was going to talk about the Lancefield stuff and all the groups. Okay, thought that was next, but it turns out no, it's in topic 8. So we'll get to topic 8 in a few minutes. Not sure if we'll get to today, but we'll come back to it and be a good review with these different Streptococcus. Kind of to finish off topic 7, I just want to talk about another thing that is sort of part of this, that culturing business and also part of microbial identification. So something that is done with a lot of microorganisms is, you can see with the Streptococcus, if I did a gram stain, they'd all look the same. So you can grow them on blood agar, and they can grow up, and so that takes a day or two. Another thing that's done with these is sometimes just looking at other metabolic capabilities. And there's literally hundreds of tests to see, basically, what do they eat, what enzymes do they have, and so on. And I'm just going to show you, I think I've got 3 or 4 tests here I'll show you that are pretty commonly used, and they're very easy to do. So first one here is the Catalase Test, and the Catalase Test is looking for an enzyme called catalase. So what does catalase do? It actually breaks down hydrogen peroxide. So you may have hydrogen peroxide in a first aid kit, and sometimes, people will get they hurt themselves, and someone pops out the hydrogen peroxide and pours it on the skin. Anyone knows what it does in your skin? It bubbles, it fizzes, yeah. And that's because your skin has the same enzyme. And so what it's doing is breaking down the hydrogen peroxide into water and oxygen gas. So this is a very easy test to do. A lot of gram positives will come up catalase positive, we call them, meaning they will form the bubbles. You can see, for example, this is actually a good test to distinguish between Streptococcus and Staphylococcus. So Staphylococcus bubbles, and I've done this test a whole bunch of times, and it's really nice. You just drop, you use a little dropper, and boom, it happens. Like, it takes 1 second, and usually it's very frothy and very quick. So this is a quick test, because of course Streptococcus and Staphylococcus are both cocci. They're very similar in size, and sometimes you don't get those nice chains or those nice clusters. So sometimes the gram stain is not going to be good enough, and you need to do some biochemical tests. So Catalase test is bubbles. So catalase positive means yes, there are bubbles, and yes, it has the enzyme catalase. Another test is something called the Coagulase test. So again, another enzyme, and this enzyme is produced by Staphylococcus aureus, but not Staphylococcus epidermidis, right? So now we're trying to distinguish between Staphylococcus. And so what it does is it has an enzyme that I'm not really sure what its biological function is, but has something to do with its breakdown and digestion of connective tissue. So a connective tissue protein called fibrinogen, right? And bottom line is, whatever it's doing, it causes clumping. And this can be seen on a glass slide, right? You can actually see it with your eyes, although this is zoomed in a little bit. So you can see what they have is a little bit of fibrinogen, and just in a dropper, so mixed in some water, and you add a little bit of the culture, and this is what it looks like if it doesn't clump, and this is what it looks like if it does clump. So like I said, there's tons of tests like this, and I'll show you one more, and we'll really go into the details on a lot of them. Here's a urease test. So again, does the organism have an enzyme called urease, which breaks down urea? A lot of these tests are you can buy kits, and they're made by companies that supply chemicals, and they've designed the kit in such a way that when it breaks down a product, you get a change in color. So in this case here, you can see the positive test, it turns red, and the negative test stays yellow. So a whole bunch of tests like this, like I said, literally hundreds of them, and this is a pretty common way for some companies to diagnose organisms without having to go through all the procedure of a gram stain and all that. Often these are very quick. They'll take seconds or 30 seconds or something like that. So in some cases, minutes. Gram stain might take a little bit longer, and you have to pull out your microscope and there's all those kind of things. There is a company, there's at least a few companies that specialize in these kits. There's one called BioMérieux, I think it's called. They're a French company, and they have thousands of these test kits. And one of the things that this company specializes in is this little, you can't really tell very maybe well from the photo, but what it is, it's a little plastic plate. And this little plastic plate, they have different versions. Some of them have 10 tests, some of them have like 50 tests. And so literally, you take the culture and you add a little bit to each little slot, I guess you'd call it, or pocket or well, and it's testing for a whole bunch of things. You can see over here, it's telling us that, okay, you know, this here, for example, ONPG, I don't know what that is code for, but ADH is alcohol dehydrogenase, LDC, I don't know what that one is, so I have to look these up, but there's urease over here. GLU is probably glucose metabolism, and then they have a code, right? And if the code says it's you know, yellow, yellow, red, yellow, blue, you know, it's Klebsiella or whatever, right? There's enough organisms that you know, like I said, they're just close enough, and you want to get that diagnosis, and you know, sometimes a gram stains on them. So like I said, you can do other metabolic tests. It's not worth it to go through all of them, but just know that that's a thing. We can test what enzymes they have and make use of their physical properties like that. Okay, so the case study, this one here, right? And we're going to talk about you know, some of these things. We have talked about everything here, and we're going to come back and you'll see we're going to talk about them again and again, and you're probably in your head. You know, not sure which organisms at the moment are gram positive and gram negative and all that. And we're going to talk about which ones to know in the next unit. But it's worth it to throw up a case study to sort of show you how these tests can be done to give a good diagnosis, right? So you can see here it says we have a skin infection, and it's floating around in a hospital. And this is MRSA. So we haven't talked about MRSA. MRSA stands for multi-drug resistant Staphylococcus aureus. This is a very serious strain of Staphylococcus aureus because it's multi-drug resistant, right? And in a hospital, you have immunocompromised people, and we have to take this very seriously. So it says here that, okay, we're not going to treat everybody, write everybody prescriptions for antibiotics. That could be pretty chaotic. But we're going to test people that are relevant, maybe people working in that wing near this particular patient, just to find out whether we should, you know, what we need to do with these people, right? So basically, swab them and see what we get, right? So I don't know if I have the words on here. I do. Okay, wasn't sure. sometimes I'm writing on the board, but I was trying to make sure everything was digital for people that are remotely. So first thing, growth on McConkey agar. And remember, McConkey agar, does it grow gram positives or gram negatives? Yeah, it grows gram negatives, right? So staph is gram positive. So that means there's not going to be any growth. So that's a quick test, right? Put it on McConkey agar. There are agars that select for gram positives only, but we haven't really discussed them. So McConkey agar is worth it to mention. What about on blood agar? We haven't actually talked about what Staphylococcus does on blood agar. We talked about what Streptococcus does. But on blood agar, Staphylococcus aureus is beta hemolytic. So that means you're going to get the clearings. And also remember that Staphylococcus aureus, it's right in the name, aureus. It has yellow colonies. Okay. So you can, that's actually two things on the agar that we can identify. The color of the colonies, yellow, that indicates Staphylococcus aureus, and the beta hemolysis is consistent with that particular organism. So gram stain, what color? For gram positive. Purple, yeah. So we're going to talk about gram stains in a minute. We've a few times already. We're going to get into more detail today, hopefully. So it's going to be purple. And that's not enough. You got to tell me what you see. It's going to be a staphylococcus, so round cells, and they're going to be clustered in most cases. The Catalase test. So remember that was a test to distinguish between strep and staph. Okay, and so staph is positive, and that means you're going to have bubbles, right? And the Coagulase test. That was the test that is going to distinguish between Staphylococcus Epidermidis and Staphylococcus aureus. And so this is positive, meaning it does clone. This is probably not all the tests that they're going to do. Probably what they will do is test the antibiotic susceptibility of this organism, because this is a multi-drug resistant version of it. So it's very easy to make some agar dishes with antibiotics in it, and you put the swabs on that. If it's growing, then you obviously have an organism that can grow in the presence of those drugs. If it doesn't grow, then the person's probably fine. Maybe they would combine all of these into one test. It's hard to tell. Kind of depends on what the administrator is deciding that they're doing with this. Okay, so that's a good introduction to, like I said, gram stains and those kind of things, which is the next unit. So that's actually, I think that's the last slide on topic 7. So I'm going to move over to topic 8. Any questions at this point? All right, so I know people want to access their midterms. Like I said, I'll make sure I look at that after the class. So please remind me, somebody can remind me right when class ends to do that, and I'll make sure I do so. Okay, so topic 8 is more on this identification theme. Like I said, I sort of broken into 2 topics. Topic 7 is more around culturing and growing these things. Topic 8 is a bit more focused on microscopy, serological methods, and DNA methods, right? So I'm hoping today we can do the microscopy stuff. Maybe not all of it, but we'll be covering and talking about gram stains for sure. So this was, if you remember my breakdown, I had these, oh, come on, there we go. That was topic 7. And remember, we went back and I showed you, I'd done some swabbing, and I showed you a demo that I had done last year, right? And these are the results that I showed you. So we have a hand, kitchen sponge, toothbrush there. And so what do we make of this, right, is kind of the question. Somebody's toes, that was my son's toes, keyboard, hand, so lots of things. I did do some swabbing after class last day. I almost forgot, but a couple students reminded me. I'll show you the results here. I took some photos this morning. So palm of the hand. Palm of the hand usually shows up pretty typical. And I can tell you just looking at the cultures that most of that is going to be Staphylococcus epidermidis. It's found on everybody's skin. Every hand I've ever swabbed, that's like 99% of the bacteria you get on there. Kind of looking at this, there's maybe one yellowish colony there, so there might be some Staphylococcus aureus there. That's nothing to be embarrassed of. A third of us probably have it on us right now, right? So it's on your skin, it's natural, it's normal. It's when it gets into other body parts that it can lead to serious things. It's hard to tell without actually doing a gram stain and other tests, but because it's not very yellow, but that would be my suspicion. Sometimes the photos don't always show up their true colors as well. Here's the light switch, and probably also a lot of other staph, but I'm also looking at a lot of these colonies are sort of flatter. And again, this is just from experience. I'm guessing there might be some bacillus there of some sort. I would have to do a gram stain to check, but you can definitely see some yellow there. This one has sort of a funny shape. I don't know what that is. I'm looking on here, something kind of white and creamy. I mean, there's quite a variety on there. Usually, it's kind of exciting. I don't usually get so much from a light switch. Usually, they're, because it's kind of a smooth surface and the bacteria